So, Alan, we've obviously seen quite an uptake within the ADC field and the buzz around it at the moment, particularly with the medchem side of things. How have you seen this evolve within the world at the moment, and how do you feel that medchem is having such an influence on the ADC field?
I think for me, what's really exciting is we're moving beyond just those standard cytotoxic payloads. We're starting to see ADCs as a delivery mechanism to facilitate the delivery of small molecules into spaces where we've not been able to go before. So, for example, molecules that have poor systemic PK or poor delivery into tissues or intolerable on-target mechanistic toxicity. Been loads of really interesting anti-cancer agents developed over the past 20 years that have not been clinically tolerated because the on-target mechanistic toxicity outweighs the therapeutic benefit. And I think they're fantastic payloads for ADCs because you can start to then repurpose or rescue those payloads, those drugs that we've not been able to get patient tolerability with, and actually make them therapeutically meaningful and get them to deliver patient benefit. But I think the understanding the medicinal chemistry is really interesting. And I think medicinal chemistry is the overlooked factor in ADC development at the moment, because understanding the nature of your payload, understanding its pharmacokinetics, understanding what happens to it after it's released from the ADC is really important. There's a lot of debate about what properties do we need to build into a payload. I mean, I think back to time spent working on respiratory programs where you want a molecule that's, for example, for asthma, delivered to the lungs and has action on the endothelia of the lungs. But then as soon as it gets into the systemic circulation, it's broken down. So you don't get that on-target toxicity. Applying that thinking to cancer therapeutics, to targeted therapeutics as part of an ADC payload, so that we start to think about, well, we have an effect on a tumor cell, but we reduce the systemic on-target toxicity because it's broken down as soon as it hits the bloodstream. So building in that metabolic instability, almost kind of switching the way we think about medicinal chemistry is really interesting, because we're now building in factors that previously we were trying to design out. So increasing metabolic breakdown, increasing potency, yes. Deciding whether we want permeability and efflux or not. Do we want to have this thing effluxed out of the cell so we get a bystander effect, or do we not want to efflux? So we want to keep it within the cell target of origin. There's a whole host of factors that we've never really considered, I think, in an ADC payload before.
So we have the main problem within the ADC field is the cytotoxicity that we often see that's off target. In the small molecule world, the main thing that we have to fight is a therapeutic window. Do you think the two worlds actually solve each other's problems? And is there something that we can build into that? Or
I think they do. I think it not only does it solve both problems, that we get the therapeutic window of the delivery and the therapeutic window of the targeted therapeutic. It almost feels like we're turning an ADC into a bispecific, but the bispecific is not a dual biological entity. It's a biological and small molecule entity. Layering in those multiple levels of selectivity gives you the ability to think about extending that therapeutic window. Not just extending the therapeutic window above and beyond what we have with the traditional cytotoxic, which we know is challenging in the ADC space, but also extending the therapeutic window potentially above and beyond what we see even with the targeted therapeutic, because we know all targeted therapeutics aren't as targeted as we perhaps might like them to be. But we also know most of them carry some form of on-target mechanistic toxicity in healthy cells anyway. So I do think there is an advantage of linking a challenging targeted therapeutic to an ADC. And it might seem counterintuitive to take something with maybe slightly challenging PK properties and attach it to a big biomolecule, which also has challenging PK properties. But our clinical colleagues know how to deal with an ADC. They know how to administer it. We can tailor the dosing schedule. We can tailor how and when it's dosed. We get better patient compliance because it's delivered in clinic.
Yeah.
So actually, I think there is an argument that we are taking two challenging independent problems, and by putting them together, we're not actually making them worse, but we're actually making them better.
Yeah, it's a really interesting thought. I mean, you mentioned linkers as well. We are thinking about building the selectivity into the payload and the antibody. But in fact, if we get the right linker as well, you've almost got three areas of selectivity that you can build into. So, I mean, do you see MedChem playing a part in both the linker development and the payload development, or is it?
I do. I mean, let's be honest, I'm a medicinal chemist by training. I'm a medicinal chemist by heart. So I'm always going to champion the role of medicinal chemistry in this. But I think understanding that design, make, test cycle and how that applies to an ADC is something that's never really been thought about before.
Yeah.
But I also get the sense that from what we've seen, from what we hear from others working in the field, there's a real sense that this is very empirical, that actually you can't optimize a small molecule and then optimize a small molecule on a linker and then just stick it on an antibody. Actually, there is a subtle, really exquisitely subtle interplay between things like your linker point of attachment, the linker length, the linker composition. And whilst you have payloads, your targeted therapeutics that may be equipotent in our enzyme, in our cell models, and they may be equipotent when you put them on a linker, conjugate them to an antibody, and all of a sudden you go from something that is therapeutically dead to something that's highly specific and highly potent. And I think it'd be great to understand that, but I do think it's still empirical. And I think that's where that expertise of the design, make, test, analyze cycle that we're so used to with small molecules comes into the ADC space, because you have to actually reduce these things to practice. You have to design and make and test them to understand which antibody is going to work in an in vivo model and hopefully in the clinic compared to those that aren't. It's perplexing, but I don't think we understand it yet. I think it's still very empirical. It's not predictable. But bringing together teams who are used to working in medicinal chemistry, in vitro cancer biology, in vivo cancer biology, and understanding, well, when you put all those bits together, it's not just like Lego. You can't bolt them together and it's a predictable outcome.
Yep.
When you put all those things together, you have to go and test it. You have to go and understand what it does in vivo.
Absolutely. And I think it's funny when you see people from the small molecule world enter the large molecule world, because it's just so different. In the large molecule world, we take this really high throughput approach and we narrow everything down, and we've got maybe 3 clinical candidates that we take through that have been screened fully with no real lead optimization to the extent of what we would do in small molecule. And the way that we've approached ADCs is a similar way. However, we're putting a small molecule onto the side of this, and we really need to think about this in the same aspect. You've got a large molecule and a small molecule, and once you've got that payload to your cancer cell or whatever your target cell is, it's going to be a small molecule at the end of the day, and we need to really build in that selectivity.
And I think it's a challenge, and it's kind of scary. And I think both communities are looking at each other going, well, I don't want to play in the small molecule space because I'm a large molecule person, and I don't want to play in the large molecule space because I'm a small molecule person.
Yeah.
But actually, I think in terms of that creativity, there's a real opportunity here, because we've got different people with different viewpoints, different experiences, different ways of looking at problems. We're not just now a small molecule community looking at small molecule problems through the lens and the blinkers that we develop over time. Similarly, for the antibody community, we're now bringing these teams together, and we're starting to think much more creatively from different perspectives about how we bring these things together. I think this whole idea of payload redevelopment in the antibody space is going to be a really interesting space to watch over the next 3 to 4 years.